Hypersensitivity, immunologically mediated tissue injury. Injurious immune reactions called hypersensitivity are responsible for pathology associated with immunologic diseases. This term arose from the idea that individuals who have been previously exposed to an antigen manifest detectable reactions to that antigen and they are therefore said to be sensitized. Hypersensitivity implies an excessive or harmful reaction to an antigen. There are several important general features of hypersensitivity disorders. Hypersensitivity reactions can be elicited by exogenous environmental antigens, that is, microbial or non microbial, or endogenous self antigens. Exogenous antigens include those in dust, pollen, food, drugs, microbes, and various chemicals. The immune responses against such exogenous antigens may take several forms, ranging from annoying but trivial discomforts such as itching of the skin to potentially fatal diseases such as anaphylaxis. Some of the most common reactions to environmental antigens cause the group of diseases known as allergy. Immune responses against self or autologous antigens cause autoimmune diseases. Hypersensitivity usually results from an imbalance between effector mechanisms of immune responses and the control mechanisms that serve to limit such responses. In fact, in many hypersensitivity diseases, it is suspected that the underlying cause is a failure of normal regulation. We will return to this concept when we will consider autoimmunity. The development of hypersensitivity diseases is often associated with the inheritance of particular susceptibility genes. HLA genes and many non-HLA genes have been implicated in different diseases. Specific example will be described in the context of diseases. The mechanism of tissue injury in hypersensitivity reactions are the same as the effector mechanisms of defense against infectious pathogens. The problem in the hypersensitivity is that these reactions are poorly controlled, excessive, or misdirected, for example, against normally harmless environmental and self antigens. Classification of hypersensitivity reactions. Hypersensitivity reactions can be classified on the basis of the underlying immunologic mechanism. This classification is of value in distinguishing the manner in which an immune response causes tissue injury and disease, and the accompanying pathologic and clinical manifestations. However, it is now increasingly recognized that multiple mechanisms may be operative in any one disease. The main types of hypersensitivity reactions are as follows. In immediate hypersensitivity, that is type 1 hypersensitivity, the injury is caused by T helper 2 cells, IgE antibodies, and mast cells and other leukocytes. Mast cells release mediators that act on vessels and smooth muscle, and pro-inflammatory cytokines that recruit inflammatory cells. In antibody mediated disorders, that is type 2 hypersensitivity, secreted IgG and IgM antibodies injure cells by promoting their phagocytosis or lysis and injure tissues by inducing inflammation. Antibodies may also interfere with cellular functions and cause disease without tissue injury. In immune complex mediated disorders, that is type 3 hypersensitivity, IgG and IgM antibodies bind antigens usually in the circulation, and the antigen antibody complexes deposit in tissues and induce inflammation. The leukocytes that are recruited produce tissue damage by release of lysosomal enzymes and generation of toxic free radicals. In cell mediated immune disorders, that is type 4 hypersensitivity, T lymphocytes are the cause of tissue injury. Immediate type 1 hypersensitivity. Immediate or type 1 hypersensitivity is a rapid immunologic reaction occurring in a previously sensitized individual that is triggered by the binding of an antigen to IgE antibody on the surface of mast cells. These reactions are often called allergy. and the antigens that elicit them are allergens. Immediate hypersensitivity may occur as a systemic disorder or as a local reaction. The systemic reaction most often follows injection of an antigen into sensitized individual, for example by a bee sting, but can also follow antigen ingestion, for example peanut allergens. Sometimes within minutes the patient goes into a state of shock which may be fatal. Local reactions are diverse and vary depending on the portal of energy entry of the allergen. They may take the form of localized cutaneous rash or blisters, that is skin allergy hives, nasal and conjunctival discharge, that is allergic rhinitis and conjunctivitis, hay fever, bronchial asthma, or allergic gastroenteritis, that is food allergy. Many local type 1 hypersensitivity reactions have two well defined phases. The immediate reaction is characterized by vasodilation, vascular leakage, and depending on the tissue, smooth muscle spasm or glandular secretions. These changes usually become evident within minutes after exposure to an allergen and tend to subside in few hours. In many instances, for example allergic rhinitis and bronchial asthma, a second late phase reaction sets in 2 to 24 hours later without additional exposure to antigen and may last for several days. This late phase reaction is characterized by infiltration of tissues with eosinophils, neutrophils, basophils, monocytes, and CD4 plus T cells, as well as tissue destruction, typically in the form of mucosal epithelial cell damage. Most immediate hypersensitivity disorders are caused by excessive T helper 2 responses, and these cells play a central role in by stimulating IgE production and promoting inflammation. These T helper 2 mediated disorders show a characteristic sequence of events: activation of T helper 2 cells and production of IgE antibodies. The first step in the generation of T helper 2 cells is the presentation of antigen to naive CD4 plus helper T cells, probably by dendritic cells that capture the antigen from its site of entry. For reasons that are still not understood, only some environmental antigens elicit strong T helper 2 responses and thus serve as allergens. In response to antigen and other stimuli, including cytokines such as interleukin 4 produced at the local site, the T cell differentiate into T helper 2 cells. The newly minted T helper 2 cell produce a number of cytokines on subsequent encounter with the antigen. As mentioned earlier, the signature cytokines of this subset are interleukin 4, 5 and 13. Interleukin 4 acts on B cells to stimulate class switching to IgE and promote the development of additional T helper 2 cells. Interleukin 5 is involved in the development and activation of eosinophils, which are important effectors of type 1 hypersensitivity. Interleukin 13 enhances IgE production and acts on epithelial cells to stimulate the mucus secretion. In addition, T helper 2 cells produce chemokines that attract more T helper 2 cells as well as other leukocytes to the reaction site.